Welcome to this explainer. Okay, I want you to imagine the trillions of cells in your body as a bustling, microscopic manufacturing plant. Inside this factory, the master blueprints, your DNA, are locked away in a highly secure vault. They're precious, they're permanent, and they're completely static. So, how does the actual work get done? Well, enter RNA. Today, we're going to explore the absolutely fascinating life cycle of this incredibly dynamic, short-lived worker that takes that static genetic code and literally brings it to life.
Let's just dive right into our roadmap for today. First, we'll meet the RNA molecule, then look at the RNA family tree, move into the transcription process, check out how we process eukaryotic RNA, and finally, look at catalysts and non-coding RNA.
All right, part 1. Meet the RNA molecule, the cell's dynamic messenger. We are going to kick off our journey by looking at its fundamental chemistry. To really appreciate what makes RNA so special, we kind of have to compare it to its much more famous cousin, DNA.
When you contrast these two molecular structures, you're going to notice two massive differences right off the bat. First up, while DNA uses a very stable deoxyribose sugar, RNA uses a ribose sugar that has a highly volatile 2' hydroxyl group. That's that little OH attached to the ring you see there. And second, RNA completely swaps out the thymine base found in DNA for a totally different base called uracil.
And honestly, this brilliantly illustrates how that extra 2' OH group completely changes the game. That little hydroxyl group basically acts like a built-in self-destruct button. Under the right conditions, it facilitates a spontaneous nucleophilic attack right on its own phosphodiester backbone, causing the RNA strand to literally cut itself in half. We call this autocleavage. But here's the thing. This chemical fragility, it's a feature, not a bug. It makes RNA a highly dynamic, temporary messenger that can be quickly degraded and replaced. That is exactly what a cell needs to rapidly respond to its environment, which is a stark contrast to the permanent, long-term storage of DNA.
Moving on to part 2, the RNA family tree, a roster of cellular workers. So now that we've got a handle on its flexible structure, let's look at its immense functional diversity. Because RNA isn't just one single thing, it's an entire cellular workforce.
The really crucial point here is the sheer variety of roles. You've got your massive messenger RNAs, or mRNAs, stretching anywhere from 1,000 to 10,000 nucleotides long. These are the actual carriers of the genetic information. Then you've got transfer RNAs, acting as these little 70 to 130 nucleotide adapters, and ribosomal RNAs that physically build the proteins. But wait, we also have these incredibly tiny non-coding RNAs, like microRNAs. These guys are barely 18 to 23 nucleotides long, and their entire job is to regulate how other genes are translated. Your cells literally rely on this entire diverse team working together to function.
Which brings us to part 3, the transcription process, copying the blueprint. Let's see how all these dynamic RNA molecules are actually born. They're created through a highly coordinated, amazing process called transcription.
But here's the major puzzle we run into. The enzyme responsible for building RNA is called RNA polymerase. When it looks at the genome, it sees billions, literally billions of base pairs of DNA. If it just started copying at random, it would be a chaotic, totally wasteful disaster. So how on earth does this molecular machinery find its exact starting line?
Well, the answer lies in specialized proteins called sigma factors. Think of a sigma factor like a highly tuned molecular GPS. It temporarily binds to the core of the RNA polymerase to form what we call a holoenzyme. This setup allows the complex to rapidly, kind of loosely, scan the DNA. When that sigma factor spots a specific genetic landmark, specifically sequences we know as the -35 and -10 boxes, it just slams on the brakes. Those sequences are the promoters. They are the precise, exact starting lines for a gene.
Take a look at the mechanics of this initiation. First, the enzyme binds to the promoter to form a closed complex. Then, it physically unwinds a small bubble of the DNA duplex to form an open complex. And what's truly mind-blowing here is that RNA polymerase can just start synthesizing the new RNA strand right from scratch. Unlike DNA replication, which absolutely requires a primer to get started, RNA synthesis is completely primer-independent. No starter needed.
So this whole birth process of an RNA molecule happens in 3 highly organized steps. First up is initiation, which we just saw. Once started, that sigma factor falls away, and the polymerase kicks into absolute high gear for step 2, elongation. It zips down the DNA, synthesizing the new RNA strand at a blazing speed of 50 to 90 nucleotides every single second. Finally, step 3 is termination. The polymerase hits very specific genetic sequences that act as a strict stop sign, signaling it to release the newly minted RNA.
All right, Section 4, Processing Eukaryotic RNA, Polishing the Rough Draft. Now, that simple system we just talked about works wonderfully for bacteria, but in complex organisms like you and me, the eukaryotes, that newly born RNA is basically just a rough draft. It needs some serious editing before it can be put to work.
The core reason for this heavy editing really comes down to cellular real estate. In simple bacteria, everything happens in one big open room. Transcription and translation, the actual reading of the RNA to make proteins, they happen at the exact same time in the exact same space. But eukaryotic cells are compartmentalized. We keep our DNA safely locked away in the nucleus, but the protein-building factories, they're out in the cytoplasm. So the RNA must be carefully processed and stabilized in the nucleus before it is ever exported.
This polishing phase involves 3 major modifications to that primary RNA transcript. Step 1, we add a protective helmet to the front, called a 5' M7 GCAP. Step 2 is splicing, where we literally cut out the non-coding interruptions in the genetic message. And finally, Step 3, we add a long protective tail of adenine nucleotides to the end, which is called polyadenylation. This cap and tail basically protect the RNA from being degraded way too early, and they help the ribosome latch on later down the line. During that middle step of splicing, a massive molecular machine called the spliceosome physically cuts out those useless introns. You can actually see it looping the intron out right there, like a lasso or a lariat. It then stitches the useful parts, the exons, together. This leaves a beautiful, continuous reading frame perfectly ready to be translated. And here's the huge takeaway. By choosing which exons to keep or skip, your cells can actually create multiple completely different proteins from just a single gene. That is incredibly efficient.
Moving to our final part, Section 5, Catalysts and Non-Coding RNA Beyond Just Messengers. To wrap up our factory tour today, we really need to shatter a massive illusion in biology. For a very long time, we thought RNA only existed as a middleman to make proteins, but it turns out that's just the tip of the iceberg. Back in 1982, a true milestone discovery completely shook the field of biochemistry, the ribozyme. This discovery proved that RNA isn't just some floppy string of information. Because it's single-stranded and can actually pair with itself, RNA can fold up into highly complex 3D structures and catalyze chemical reactions exactly like a protein enzyme. They can slice other RNA molecules apart or splice them together. RNA can absolutely do the heavy lifting of cellular chemistry all on its own. And when we finally map out the human genome, the sheer scale of RNA's other jobs becomes stunningly clear. Look at how tiny the slice is for protein coding. A shockingly massive proportion of our DNA is actually transcribed into non-coding RNA. For decades, scientists thought this was just useless transcriptional noise. But actually, scratch that. It is definitely not noise. These long and short non-coding RNAs act as structural scaffolds, they recruit proteins, they silence viral invaders, and they strictly regulate exactly how our genes turn on and off. They are the true managers of the cell. Given all these incredible dynamic capabilities, it really leaves us with a fascinating evolutionary question to ponder. We know DNA stores information, right? And proteins do the chemical work. But RNA can do both. It can carry genetic code, and as ribosomes show us, it can catalyze chemical reactions. Could this incredibly versatile, hard-working, self-cleaving molecule actually be the original molecule of life, right at the very dawn of biology? It's a profound thought to leave you with. Thanks so much for joining me on this explainer, and keep questioning the incredible machinery inside you.